smartphone. So we used to have lots of time, and that time we used to use for chatting with friends and read books. And one of the books during my MSc days that was of interest is this one called Junk DNA. Okay, and the reason being that only 2% of any eukaryotic genome, higher organisms, including human, is coding for proteins, and rest 98% is not doing any coding function. So they are. That's what people used to think in our times. And several books came, and over 15 years, people started questioning what's in your genome, and then the myth of junk DNA, that probably this concept of junk is not right. So as I was telling you, in our chats, during our when some of the friends used to assemble together, we used to discuss what is this junk DNA. I'm talking about early 90s, my MSc days. So is it the dark matter of the genome? I mean something very important, just we do not know what is doing. And the idea is that if it is very important and we just don't know what it is doing, just don't call them junk. Just I do not understand something doesn't mean that it's junk. That is the idea. So back in 2000, these were the questions people were asking, that only 2% of the human genome were coding for proteins. Rest 98% were not doing, and more than 90% of the genome is actively transcribed. Even though it is not coding for protein, it is giving rise to RNA, and something is happening. So why? Because in living system, nature doesn't do anything that is wasteful. Just like that, nature will not keep 98% of genome doing nothing, and still it will be transcribing into RNA, wasting a lot of energy. This is not acceptable at all in natural science. So what is it? And in another 20 years, people are asking that this nomenclature of junk is actually extremely misleading. This is not correct. And the question now is what exactly they are hiding. Okay? What is in there, in the junk part of any living system, higher organisms? So before I go into the topic, when molecular biology started long back, almost 80 years back, and you know who started it? Do you have any idea? I am assuming most of you are students, so you might be having idea. So the concept of molecular biology is given by a physicist named Erwin Schrödinger. He actually proposed what is life. And if you look at the Nobel laureates, 80% came from physics or chemistry background. Okay? So that time, some ideas came, and over the years we noticed that there are at least 3 issues that are haunting. What are these root problems? Number one, we know that there is something called central dogma of molecular biology, that DNA to RNA to protein, this is the way information flows. Okay, this we know. But over time, RNA viruses came, and just last 2 years we have seen what an RNA virus can do. And people modified this, that there could be information flow in the opposite direction as well, RNA to DNA, and then going forward. But in last 20 years, this dogma has been completely modified. It has changed its look because this new player that has come into the business called non-coding RNA, which comes from the 90% of the genome which erstwhile people used to call the junk, and I will be centering my talk about this non-coding RNA. And people realize that information flow and regulation can occur in all possible directions. It's no longer unidirectional as we used to think, as people used to think. Okay? So this is the problem number 1 in modern biology. Second problem is kind of offshoot of that protein is the endpoint. So they are the structural functional molecule. So protein must have a fixed structure, and through that fixed structure, it will do its function. So this is called the structure function, sequence structure function paradigm. And in last 20 years, this paradigm is heavily challenged. People are asking all sorts of questions about it. So what is this second issue, second problem? 3 examples I will put in front of you. Number 1, this is a classic Nature paper, 1996, by these 2 guys. What they did, they took same protein called protein G, it has a fixed structure, and they took this 11 residue amino acid stretch and put it in some other place in the same protein. So if they replace this stretch and they took this protein stretch and put it there, synthetic, right? Recombinant DNA technology, and found that within the same protein fold, here it is an alpha helix and here it is an extended coil. So that means that sacrosanct rule of sequence is giving you fixed structure is no longer true. That is number 1. This is called the chameleon sequences. That was discovered back in 96. Then came the moonlighting proteins. These are very interesting. And first part of my career as a faculty member, I spent time on this because all important proteins in our body, many of you who are in biology have heard the name of P53, the tumor suppressor protein. They interact with so many other proteins. For P53, the number is around 450. And so, if it has to interact specifically with these many interactors, it has to have a huge surface area. That means you are talking about a big protein. That means you are talking about molecular crowding inside the cell. So people figure out that that is not how they work. They are kept as a open chain, and when the particular interactor comes, they fold accordingly. So these are called moonlighting proteins. Example I have given, a very common example, lactate dehydrogenase. We know how it takes part in the pyruvate lactate conversion, right? But the same protein in the dark eye is called epsilon histidine, exactly same protein, okay? And it has same sequence and structure, but it does completely different function in a different organism, in a different tissue, okay? So these are called moonlighting proteins. That means a protein which can do different functions. And the third example is called promiscuous enzymes. Promiscuous means somebody who is having multiple partners. Okay. So this is an example of a hydrolase. And what does hydrolase do? They take water molecule to cleave another protein. Okay. But by 2006, people figured out that hydrolase can actually take part in all sorts of chemistry. Aldol condensation to classic epoxidation and everything. So the term hydrolase is no longer fixed. It can interact with many other partners, and it can do differently depending on the biological context. So the point is that paradigm of sequence giving rise to a fixed structure which is doing a particular function is no longer true. That is the second issue. And the third problem, so this is the statement, and evolutionary confirmation is not always essential for function. I will come back to this point at the end of my talk. And the redundance enigma. This is a very old problem which is called the C-value paradox. When people started understanding the entire genome of big organisms, they thought that the increase of genome size is somehow correlated with the complexity of the organism. When they plotted this, so different organisms versus the genome size, so plotted, and they found there is absolutely no correlation. Okay, so much so that mammals, which we think are the superior animals, are here, and there are many here, and there are many other organisms like amphibians. They have even bigger genomes. So the complexity, genome size, and number of proteins, they are not absolutely correlated. They are not doing such kind of correlation at all. So this is called C-value paradox, and this is now also explained with the help of the jump thing that I am going to do. So in the genomics era, the basic pillars of molecular biology are being posted. Now, before I go into step into the genomics era, two examples of pre-genomic era I would like to put in front of you. Number one is the discovery of the first linked RNA. This is the topic of today's lecture. This is called H19. So back in late 80s, people actually discovered this linked RNA, H19. But they didn't know the term linked RNA. Actually, when I did my PhD and completed my postdoc, came back as a faculty in Shanghai Institute, this term was not there. So it came only in 2009. This is very recent, and the research in linked RNA has started only in 2012. So they called it an mRNA, H19 mRNA. Okay, so it was discovered much before. And another milestone discovery we have in our lab, we have special interest in it called cyst. So this particular linked RNA was discovered in 1989, and it has role in X chromosome inactivation. Those from zoology background, they know that in female there are two X chromosomes, and for dosage compensation, one must be silenced, and that process is somehow regulated by this linked RNA cyst. I'll come to this briefly. So then comes the genomics era. This is a picture of Bill Clinton in White House declaring that the chromosome 22 of human is completely sequenced. You have heard about it, Human Genome Project. Okay? And this is Greg Venter and this is Francis Collins, who are sitting beside two scientists who are the leaders in this project. And obviously, after this, 2000, all the papers declare that we have stepped into the post-genomic era and biology is going to change after that. How? We are going to discuss. So, whatever has happened, the biggest contribution of Hugo was new technologies that came in the market. In 2009, this technology, which is called next generation sequencing, was very much abundant. And you know, the only technique by which you can distinguish between a delta variant of COVID and Omicron variant is this technology. So this is what is happening now. The point is, this technology helped us in sequencing the entire genome. Not only the coding regions, but the 98% of the rest, the non-coding regions as well. So that helped us understand what is in there in those junk areas and what exactly they are doing functionally. So, how do they do that? There are many ways. So, I'll discuss. So, but what happens, this technology, it can sequence entire chromosome and then one can actually use computational tools, high throughput pipelines to figure out that what are these in-between regions between the genes, okay? And then they can correlate those with the function. So we have started understanding what this dark matter is all about, okay? But have we understood them yet? No. We have just started doing this technique, using this technique. Now the question that comes in mind that these linked RNAs, which are sitting in between the coding genes, how do you know that there is a link? Good thing is that most of the linked RNAs are linked with histone modification mark. You know histone, that those basic proteins which help form the chromatin, and it gets modified at specific amino acid locations. Lysines are modified with methylation or acetylation and things like that. So one can correlate the modification marks of the histones and specific motifs present in the linked RNA, by which one can actually identify whether this sequenced region is a linked RNA or not. So these are the ways by which we understand whether this is a linked RNA or something else. Okay. What happened actually, this technology helped us to delve deeper into the junk, so-called junk, which 20 years back people used to think as junk DNA. Now we can look in a deeper way, and what people have realized that the human genome is permissibly transcribed. I mean, if you look at this chart, only 2% of the human genome is transcribed to form certain proteins. Rest 90% give rise to, they are also transcribed. And some other portion, about 7 to 8% remains, which is basically the regulatory regions, okay, promoters, enhancers, and things like that. Whatever it is, these non-coding RNA are of two types, circular RNA and linear RNA. And they can be short non-coding RNAs, of which microRNA almost all of us have heard about. They are small 22 nucleotide regulatory molecules. There are many other such small RNAs, and similarly there are long non-coding RNAs, something of our interest, and they can be sense, intronic, and many other types. I'll discuss in detail. So the point is, the ones ignored and jumped vastness. Why vastness? Because 98% was unknown to us. Now we know that we cannot ignore them and just jump them. So now it had more meaning than we could fathom. In the C-value paradox, we talked about how it is not correlated with the size of the genome, and now we can understand how complexity arises. The non-coding RNAs, which are any protein, help forming any protein, are actually doing the regulation, whether a gene is going to be expressed or not, and all those important things are being controlled by them. So they are also called regulatory RNAs, and answer to the genome complex. Now, question is, how are they generated, and what is the fate of this linked RNA inside the cell? How are they generated? Just like how exactly mRNA is generated. So you have the linked RNA region, and so it is getting transcribed. It may be degraded also, or it may just get adhered to the chromatin. If you remember, I said that identification of a linked RNA is usually done by the chromatin modification markers, so it can be just attached to that. Or many other things can happen. It can help it can be spliced also in incomplete splicing, and many other proteins can get associated. It can come out of the nucleus just like an mRNA, and it can get associated with other proteins, associated with ribosome. Then it can get degraded. It can go inside organelles, like in this case this is shown inside the mitochondria, or it can go inside exosomes and things like that. Okay, so it looks like long linked RNA, although they are smaller in number, they are present everywhere inside the cell, and they can do many things. What are the things that they can do? Before I talk about the function of linked RNAs, there are various linked RNA species. They can be just like mRNA, they can be transcribed along the reading frame, they can be transcribed just opposite to the reading frame, okay? And they can be associated with some other protein, they can be capped, they have poly-A tail, and just like mRNA and things like that. So accordingly, many linked RNAs have been identified by scientists in the last 20 years. So, as I said, these are the functions of linked RNA. They can be associated, they can act as an enhancer. So here, this red one is a linked RNA. It is attaching with a chromatin, and then it is attaching with another nearby chromatin complex. And as a result, it can pull this DNA strand together so that the local concentration of these proteins increases just in upstream of a gene, so that this gene is transcribed in a better way. Similarly, it can help modify the chromatin, okay? Pull in the modified enzymes near the chromatin and make methylation and acetylation possible. And it can do so many other things. Most importantly, what these linked RNAs do, it has small motifs through which it can bind microRNAs and act as a microRNA decoy or sponge. Means microRNAs are small regulatory RNAs that can go and bind somewhere and stop gene expression, increase gene expression. Now, if linked RNAs pull the microRNAs back, then they cannot act normally, so that the gene expression is sparked out. So this is called sponging of microRNA by linked RNA. That this is some function that we are interested in the lab. So this was what I was discussing. They can act as a decoy by which it can bind to multiple microRNAs and stop their normal function. It can act as a scaffold, dynamic scaffold, on which link RNAs sit, and then it brings in those proteins on the genome and help in the chromatin remodeling and this kind of functions. This. And as I told, this sponging action of link RNAs, that when they are present in higher abundance, they can actually hold similar microRNAs, several microRNAs, and stop their normal function. So this is one such microRNA that actually acts this way. So, in summary, what do link RNAs do? First, link RNA regulate gene expression at different levels, pre-transcriptional, transcriptional, as well as post-transcriptional level. And it, as I said, it can act as guides, decoy, sponges or scaffolds to regulate gene expression. It can increase expression of a gene or decrease. And this way, link RNAs are associated with various diseases. Most of the link RNAs are, work have been done on cancer, but in cardiovascular disease, neurodegeneration also, people have seen the link RNAs are playing very important role in controlling the disease. So people are thinking as whether instead of protein, whether one should work on link RNA as potential biomarker. Now question is, these are very small in number, unstable, and they do not have a fixed structure. Okay? So how do you study those? So people have devised certain methods and continuous upgradations are going on. So this is called chromatin cross-linking and immunoprecipitation. Biologists might have already heard about immunoprecipitation. So what you do, you are going to catch hold of a protein, so you raise an antibody against it and pull down the entire antibody and protein complex. Okay? So here you do the same thing. Only additional thing is that your link RNA is attached to it, so you can study it. Similarly, chart can be done, which is chromatin immunoprecipitation and entire chromatin. So the DNA, protein and the RNA, everything is pulled down and then you separate them and study them individually. So these are pretty standardized techniques now. What is not very standardized is this, which is called shape mapping. This gives you structural information of the link RNA. Link RNA do have secondary and tertiary structures, and that can be determined by chemical mapping. So RIP is one of the more popular technique, RNA immunoprecipitation. So what you do, you have the RNA circulating in the biological system, you know which protein it interacts with, you have an antibody, you capture the protein with that antibody, pull everything down so that your RNA is going precipitating down and you isolate and study that. So this is an easier technique. This I already talked about. Question is, can RNAs be captured directly? If you remember the techniques that I mentioned right now, all of them are, you are going to capture the RNA via somebody else, like a protein you are capturing or the chromatin you are capturing. Can they be captured directly so that you study them directly? Yes, there is a method. And in India, we are trying to kind of standardize this technique. Nobody has done it. In 2016, this was proposed, the technique, where you have the entire complex, where you have the DNA, some proteins which are binding to DNA, as well as the link RNA which is holding onto it. So what you do, you design probes which is biotin related. You know biotination is a marker. And so that these small probes, they go and attach to the link RNA, and then you pull down this link RNA directly through strong streptoadenine pulling down process, and then you separate the DNA, RNA and protein and do this DNA sequencing or RNA sequencing or mass spectrometry kind of thing. So this is a very robust technique we can standardize. So, everybody has a right to be skeptical, because for last 100 years we are talking about proteins and proteins and proteins only. DNA came only very recently, last 50, 60 years. So, question may be why link RNA and not protein or small RNA? Number one, unlike proteins and small RNAs, link RNAs are tightly tethered to chromatin in 6. That means this is the only molecule which can give you direct hint about allelic regulation. Second, they have unique address. Address is more than just a sequence motif. A small RNA or protein can identify a sequence motif, but it can identify a location, compartment inside the cell, and a complex. So much more than that. And it can exert a variety of functions via its secondary and tertiary structure. So knowing this, one can actually get much more biological information than what just a protein or a small RNA can give you. So that's why most of the big labs worldwide, they are focusing on understanding. So what are the future directions? This is not end, by the way. Future directions is, there are several questions. I have just picked up a few. For example, does link RNA bind to chromatin complex promiscuously? Means you just add a link RNA, it goes and binds to any site of the chromatin, or it has some kind of specificity. No one knows that. What is the biogenesis pathway? It's not specifically known. What triggers its generation and how the partial splicing takes place, etc. Link RNA function, this is our interest. New tools for analysis and many other questions can be there. So I will give you an example of this thing, the question that we are trying to address, link RNA function in vivo and disease and development perspective. What function do they do and how can they be correlated with diseases? In our case, these are neurodegenerative diseases like Alzheimer's disease. But the most important take-home message is what I just now said. In the last about 100 years of molecular biology, we have learned most of the things about this. They are the most prevalent entity in the cell. You just break open a cell, it's full of proteins. But their information is stored in only 2% of the genome. Rest 98% are not coding for protein, of which 90% is coding for these link RNAs. Okay? So the next 50 years of biology could be spent to learn what the rest of the entities do, whose information is stored in 90% of the genome. This is not easy because these molecules are not easy to track, they are unstable, and their structures are highly flexible. But this is where biology is going in next 50 years. So what we do in the lab? First of all, by training, I am a protein crystallographer, I am a protein biologist. I did, all throughout my life, I worked with proteins. So far, out of 16, 10 of my PhD students have worked on protein only. Okay? So, but there are certain interesting things. First of all, neurodegeneration like Alzheimer's disease and cancer, they are like, complete antidote to each other. They don't coexist together. So in recent times, there are hypotheses that Alzheimer's disease and cancer, two monsters cannot be staying together. Either a person will be having cancer or Alzheimer's disease. Good for us, because if the person had both, then our life would have been more complicated. Okay? But the interesting thing is that link RNA and disease database reports about 5,000 odd Alzheimer's associated link RNAs, whereas for cancer the number is about almost 90,000. You know why? Because the research started in 2012 and since 2015, in last 7-8 years, almost entire work on link RNA is done by cancer biologists. Neuroscientists have just picked up the subject. So this is the difference in number. Interesting thing is that almost all of these 5,000 Alzheimer-related linked RNAs also belong to cancer. Does that mean that these linked RNAs are omnipotent? They can take part in just two different kinds of events, cellular events, cancer being the proliferative event and neurodegeneration being a degenerative event. Same linked RNA taking part in both the events. Very promiscuous kind of action. And the third thing is what these linked RNAs, which are known to be involved in neurodegeneration, what do they have in common? So this is what we have been doing in the last 5 years. First, my student Kaushik, what he did, he using an Alzheimer's disease mouse model, he identified all the non-coding, note this point, non-coding transcriptome. Transcriptome means whatever is transcribed from the DNA. So it would be microRNAs and linked RNAs as well. So these are the microRNAs that we discovered which are involved in Alzheimer's disease. Here red means they are upregulated and green means they are downregulated in Alzheimer's. So he identified all these in Alzheimer model, mouse model. Similarly, he found there are 41 deregulated linked RNAs in Alzheimer mouse model. Okay. Immediately I asked him, in my lab we work with human systems, we work with human cells and things like that. So what these 41 linked RNAs are going to do? So you identified how many of these have human homologs, and he figured out there are 17 human homologs. And then what he did, started validating these 7 AT-associated linked RNAs in the disease models, Alzheimer's disease tissue, human tissue. So you don't kill a human being. After death, this is post-mortem brain. You collect from Alzheimer's patient, after their death. So and he figured out all those which he identified in mouse are also, up or down regulated in Alzheimer's disease. And this is a very short. This is a very short, busy slide. Here we identified their location also in certain punctate structure inside the nucleus. We have very high super resolution microscopy. We could see what exactly these linked RNAs are doing, associated with whom, in which compound. And then one by one my students started elucidating the linked RNA functions and which microRNAs they are sponging into. For example, MALAT1, it was predicted by bioinformatics that it binds to these 3 microRNAs, and then Kaushik showed that they are really getting bound, to the MALAT1 linked RNA. And what happened? MALAT1 can bind to these microRNAs, but the opposite is not going to happen. So if you get rid of these microRNAs, the linked RNA, nothing happens to the linked RNA. So it's an unidirectional sponging effect that is established. This paper has just accepted. Then the second linked RNA called NEAT1, and he figured out that these 2 microRNAs are regulated by NEAT1, and then these microRNAs go and are involved with certain signaling proteins for decades. Then another student is involved in doing certain bioinformatics. This is what he did during the lockdown period. He was sitting in home, so I asked him to do this research. So he is from zoology background, MSc zoology from Pondicherry University. So he learned coding, computer coding. Okay, learned C++, Python, and R package, and wrote this code and figured out that all those 500 Alzheimer-related 5,000 Alzheimer-related linked RNAs, they are functionally clustered in several groups, and figured out some important members from these groups, and started him, by that time lockdown was over, he could join the lab back and went back to the lab and started validating their role in the Alzheimer's process. So he found out that they are upregulated, severely upregulated in Alzheimer model, And this cyst, as I said, is involved in X chromosome inactivation. So these are different patient samples, trisomy and others, and how their concentration increases because of the multiple copies. So I think I should stop here. This is our beautiful auditorium in Shanghai Institute of Nuclear Physics. I figured out that Shankar is also going to show you the similar kind of pictures. But anyway, and we are affiliated to Shanghai Institute of Nuclear Physics, and HBNI is our university with which we are affiliated to, and we are generously funded by DAE and DST. And the main players of this new RNA project are Kaushik Chandra, who just completed PhD last year and now in Swiss Institute of Neurology,